When I went to college, I had to buy this biology book, and we learned about almost every chapter except for the one on viruses and metabolism. And there's just some things I want to point out because I feel like everyone should know this, and because people need to read up on viruses and figure out, if all of these things are telling us is true. It says, recall that bacteria and other prokaryotes are cells much smaller and more simply organized than the cells of eukaryotes, such as plants and animals. Viruses are smaller and simpler still. So viruses are smaller than bacteria and animal cells, if you didn't already know. It says, early on, they were considered biological chemicals. In fact, the Latin root for the word virus means poison. And down here it says, beyond their value as experimental systems, viruses have unique genetic mechanisms that are interesting in their own right and that also help us understand how viruses cause disease. In addition, the study of viruses has led to the development of techniques that enable scientists to manipulate genes and transfer them from one organism to another. These techniques play an important role in basic research, biotechnology, and medical applications. For instance, viruses are used as agents of gene transfer in gene therapy. And there's this guy, and his name was Mayer, and he discovered viruses, and he said, he's pretty much saying that diseases caused in the sap of plants was caused by unusually small bacteria that were invisible under a microscope. So they're so small you can't even see them under a microscope. And it says, so you have to use a thing called an electron microscope, which means bacteria or viruses are smaller than bacteria and animal cells. The tiniest viruses are only 20 nanometers in diameter, smaller than a ribosome. Millions could easily fit on a pinhead. Even the largest known virus, which has a diameter of several hundred nanometers, is barely visible under the light microscope. So a virus is smaller than a ribosome, and a ribosome is the smallest organelle in an animal cell. An organelle is like an organ in a human body, only in comparison to a cell. And a ribosome is the smallest organelle in an animal cell. And it's saying that viruses are smaller than even this. And the reason surgeons wear masks when they're doing surgeries on people is to prevent them from getting infected by bacteria, which is hundreds of times bigger than a virus. And then it goes on to say, Stanley's discovery that some viruses could be crystallized was exciting and puzzling news. Not even the simplest of cells can aggregate into regular crystals. But if viruses are not cells, then what are they? Examining the structure of a virus more closely reveals that it is an infectious particle consisting of nucleic acid enclosed in a protein coat, and for some viruses, surrounded by a membranous envelope. And it talks about viral genomes, and some viruses can be DNA and others can be RNA, and there's double and single stranded.
Adenovirus capsid and envelopes. Adenoviruses, which infect the respiratory tract of animals, have 252 identical protein molecules arranged in a polyhedral capsid with 20 triangular facets in icosahedron. Thus these and other similarly shaped viruses are referred to as icosahedral viruses. An adenovirus has a helix shape, and it's got 6 protein spikes, glycoprotein spikes, at each vertex. An influenza virus has an outer envelope studded with glycoprotein spikes. The genome consists of 8 different RNA molecules, each wrapped in a helical capsid. And there's rod-shaped viruses that have helical capsids, overall shape of a rigid rod. A bacteriophage, T4, like other T-even phages, has a complex capsid consisting of an icosahedral head and a tail apparatus. Some viruses have accessory structures that help them infect their host. For instance, a membranous envelope surrounds the capsid of influenza viruses and many other viruses found in animals. And going on, bacteriophages or phages. The first phages studied included 7 that infect E. coli. These 7 phages were named type 1, T1, type 2, T2, and so forth in the order of their discovery. And phages infect bacteria. Viruses replicate only in host cells. Viruses lack metabolic enzymes and equipment for making proteins, such as ribosomes. They are obligate intracellular parasites. In other words, they can replicate only within a host cell. So, it's saying viruses are parasites. Furthermore, viral infection of a multicellular eukaryote is usually limited to particular tissues. Human cold viruses infect only the cells lining the upper respiratory tract, and the AIDS virus binds to receptors present only on certain types of white blood cells. And this is showing the replicative cycle of a viral cycle. A virus is an obligate intracellular parasite that uses the equipment and small molecules of its host cell to replicate. In the simplest of viral cycles, the parasite is a DNA virus with a capsid consisting of a single type of protein. And this one uses messenger RNA. After the viral nucleic acid molecules and capsomeres are produced, they spontaneously self-assemble. Alright, hold on. Down here it says many DNA viruses use the DNA polymerases of the host cell to synthesize new genomes along the templates provided by the viral DNA. In contrast to replicate their genomes, RNA viruses use virally encoded RNA polymerases that can use RNA as a template. So these viruses can inject RNA into the host cell and use it as a template to basically turn it into messenger RNA. It says, after the viral nucleic acid molecules and capsomeres are produced, they spontaneously self-assemble into new viruses. In fact, this gets interesting. Researchers can separate the RNA and capsomeres of TMV, tobacco mosaic virus, and then reassemble complete viruses simply by mixing the components together under the right conditions. So it's saying they can make viruses. The simplest type of viral replicative cycle ends with the exit of hundreds of thousands of viruses from the infected host cell, a process that often damages or destroys the cell. Such cellular damage and death, as well as the body's responses to this destruction, cause many of the symptoms associated with viral infections. We will now take a look at some of these variations in bacterial viruses, phages, and animal viruses. Later in the chapter, we will consider plant viruses. And this is the replicative cycle of a phage, bacteriophage. And it talks about the lactic cycle. A phage replicative cycle that culminates in death of the host cell is known as a lactic cycle. The term refers to the last stage of infection during which the bacterium lysis breaks open and releases the phages that were produced within the cell. Each of these phages can then infect a healthy cell, and a few successive lactic cycles can destroy an entire bacterial population in just a few hours. A phage that replicates only by a lactic cycle is a virulent phage. So it's talking about how viruses can destroy bacterial cells. So why are we using germ-killing stuff on our hands to kill bacteria? It's just crazy because phages kill bacteria, and it can disrupt the balance of your microbes. After reading about the lactic cycle, you may wonder why phages haven't exterminated all bacteria. In fact, phage treatments have been used medically in some countries to help control bacterial infections in humans. Bacteria are not defenseless, however. First, natural selection favors bacterial mutants with receptors that are no longer recognized by a particular type of phage. Second, when phage DNA successfully enters a bacterium, the DNA often is identified as foreign and cut up by cellular enzymes called restriction enzymes, which are so named because their activity restricts the ability of the phage to infect the bacterium. The bacterial cell's own DNA is methylated in a way that prevents attack by its own restriction enzymes. So the cell has enzymes that destroy its own DNA. It's methylated. so that so that it can't infect other cells. But just as natural selection favors bacteria with mutant receptors or effective restriction enzymes, it also favors phage mutants that can bind the altered receptors or are resistant to particular restriction enzymes. Thus, the parasite-host relationship is in constant evolutionary flux. Instead of lysing their host cells, many phages coexist with them in a state called lysogeny, which we'll now discuss. The lysogenic cycle. In contrast to the lytic cycle, which kills the host cell, the lysogenic cycle allows replication of the phage genome without destroying the host. Phages capable of using both modes of replicating within a bacterium are called temperate phages. So it's saying viruses can exist within the cell and replicate in a normal way with the cell without destroying the cell. So you can get infected by a virus and have no symptoms. And it says, during a lytic cycle, the viral genes immediately turn the host cell into a producing factory, and the cell soon lyses and releases its viral products. During a lysogenic cycle, however, the aDNA molecule is incorporated into a specific site on the E. coli chromosome by viral proteins that break both circular DNA molecules and join them to each other. When integrated into the bacterial chromosome in this way, the viral DNA is known as a prophage. One prophage gene codes for a protein that prevents transcription of most of the other prophage genes. Thus, the phage genome is mostly silent within the bacterium. Every time the E. coli cell prepares to divide, it replicates the phage DNA along with its own and passes the copies on to daughter cells. A single infected cell can quickly give rise to a large population of bacteria carrying the virus in prophage form. This mechanism enables viruses to propagate without killing the host cells on which they depend. The term lysogenic implies that prophages are capable of generating active phages that lyse their host cells. This occurs when the A genome is induced to exit the bacterial chromosome and initiate a lytic cycle. An environmental signal, such as a certain chemical or high energy radiation, usually triggers the switchover from the lysogenic to the lytic mode. So it's saying environmental factors or contaminants can switch this viral cycle from a lysogenic to that doesn't kill the cell and replicates with the cell to a lytic cycle that destroys the cell. In addition to the gene for the transcription preventing protein, a few other prophage genes may be expressed during lysogeny. Expression of these genes may alter the host phenotype, a phenomenon that can have important medical significance. For example, the 3 species of bacteria that cause the human disease diphtheria, botulism, and scarlet fever would not be so harmful to humans without certain prophage genes that cause the host bacteria to make toxins. And this is a table, and classes of animal viruses. As you can see, adenovirus is a double-stranded DNA. It has no envelope, and it's a virus capable of causing respiratory issues and tumor-causing viruses. So respiratory viruses and tumor-causing viruses. Single-stranded DNA, single-stranded RNA serves as messenger RNA. You can see coronavirus is in this part of the table, and it's saying that coronavirus is a single-stranded RNA that creates messenger RNA inside of the cell. And it does this by using what I mentioned earlier, virally encoded RNA polymerases that can use RNA as a template. And retrovirus, single-stranded RNA template for DNA synthesis. And a retrovirus does have an envelope, and so does the coronavirus. It causes severe acute respiratory syndrome. But a retrovirus has an envelope, and one example of a retrovirus is human immunodeficiency virus, HIV or AIDS. And not only that, an RNA tumor virus or leukemia. So it's saying that retroviruses can cause cancer and leukemia and tumors as well, because they destroy your immunity. They destroy your immune system. That's what HIV does. It destroys your immune system. Replicative cycles of animal viruses. Many variations on the basic scheme of viral infection and replication are represented among the animal viruses. One key variable is the nature of the viral genome. Is it composed of DNA or RNA? Is it double-stranded or single-stranded? Single-stranded RNA viruses are further classified into 3 classes, 4 and 5, according to how the RNA genome functions in a host cell, whereas few bacteriophages have an envelope or RNA genome. Many animal viruses have both. So it's saying that many animal viruses have envelopes and RNA genomes. In fact, nearly all animal viruses with RNA genomes have an envelope, as do some with DNA genomes. And it says RNA as the genetic material of many animal viruses. So, go back to this. Retroviruses are single-stranded RNA. Coronavirus, single-stranded RNA. And it's saying many animal cells, RNA is the genetic material of many animal viruses. So, viral envelopes. In a process much like exocytosis, new viral capsids are wrapped in membrane as they bud from the cell. In other words, the viral envelope is derived from the host cell's plasma membrane. Although some of the molecules of this membrane are specified by viral genes, the enveloped viruses are now free to infect other cells. This replicative cycle does not necessarily kill the host cell, in contrast to the lytic cycle of phages. In the case of herpes viruses, copies of the viral DNA can remain behind as mini chromosomes in the nuclei of certain nerve cells. There they remain latent until some sort of physical or emotional stress triggers a new round of active virus production. RNA as viral genetic material. Although some phages and most plant viruses are RNA viruses, the broadest variety of RNA genomes is found among the viruses that infect animals. Among the three types of single-stranded RNA genomes found in animal viruses, the genome of class 4 viruses can directly serve as messenger RNA and thus can be translated into viral protein immediately after infection. So class 4 is coronavirus. Class 4 is coronavirus. And it's saying that the genome of class 4 viruses can directly serve as messenger RNA and thus can be translated into viral protein immediately after infection. And the jab uses messenger RNA in a different way. All viruses that require RNA synthesis to make messenger RNA, use a viral enzyme capable of carrying out this process. There are no such enzymes in most cells. The viral enzyme is packaged with the genome inside the viral capsid. The RNA animal viruses with the most complicated replicative cycles are the retroviruses. So it's saying the RNA animal viruses with the most complicated replicative cycles are the retroviruses, class 5. These viruses are equipped with an enzyme called reverse transcriptase, which transcribes an RNA template into DNA, providing an RNA to DNA information flow, the opposite of the usual direction. This unusual phenomenon is the source of the name retroviruses. Retro means backward. So it takes RNA and turns it into DNA instead of DNA ultimately creating RNA. And this is a replicative cycle of enveloped RNA virus. And you can pause it if you need to. And of particular medical importance is HIV, immunodeficiency virus, the retrovirus that causes AIDS, acquired immunodeficiency syndrome. HIV and other retroviruses are enveloped viruses that contain two identical molecules of single-stranded RNA and two molecules of reverse transcriptase. Figure 19.8 traces the HIV replicative cycle, which is typical of a retrovirus. After HIV enters a host cell, its reverse transcriptase molecules are released into the cytoplasm, where they catalyze synthesis of viral DNA. The newly made viral DNA then enters the cell's nucleus and integrates into the DNA of a chromosome. The integrated viral DNA, called a provirus, never leaves the host genome, remaining a permanent resident of the cell. Recall that a prophage, in contrast, leaves the host genome at the start of a lactic cycle. The host RNA polymerase transcribes the proviral DNA into RNA molecules, which can function both as messenger RNA for the synthesis of viral proteins and as genomes for the new viruses that will be assembled and released from the cell. In Chapter 43, we describe how HIV causes the deterioration of the immune system that occurs in AIDS. And this is the replicative cycle of HIV. But it's crazy because it's saying that retroviruses, which are the most complicated, have the most complicated replicative cycles, and there's no cure for HIV. So that tells us that there's a lot of stuff we still don't know about retroviruses, and that they come from animal cells, which are used to make vaccines. And they have they integrate into the viral DNA and never leave the host genome. So they never leave the genome of the cell. And that's how they that's how they deteriorate your immune system. You would never know that you was infected by it because it stays in your cell. Other viruses, viroids, and prions are formidable pathogens in animals and plants. So now it's talking about viroids and prions. It says other smaller or less complex entities known as viroids and prions also cause disease in plants and animals, respectively. Viral diseases in animals. Viruses may damage or kill cells by causing the release of hydrolytic enzymes from lysosomes. Some viruses cause infected cells to produce toxins that lead to disease symptoms, and some have molecular components that are toxic, such as envelope proteins. And then it talks about a vaccine is a harmless variant of a derivative of a pathogen that stimulates the immune system to mount defenses against the harmful pathogen. But the thing is, a vaccine wouldn't work against a retrovirus because it surpasses the immune response, because a retrovirus is within the genome of a cell. And they're given all these, shots, and it's like overstimulating the immune system. I mean, against the coronavirus, against the flu, against, Because remember, a retrovirus is a provirus. It's silent. So it says the antibiotics that help us recover from bacterial infections are powerless against viruses. So it's saying antibiotics are powerless against viruses. So why are we using germ-killing hand sanitizer and all these antibiotics to kill viruses when antibiotics don't kill viruses. And antibiotics kill bacteria by inhibiting enzymes specific to bacteria, but have no effect on eukaryotic or virally encoded enzymes. However, the few enzymes that are encoded by viruses have provided targets for other drugs. And then it talks about other treatments that you can get besides vaccines. It says most antiviral drugs resemble nucleosides and as a result interfere with viral nucleic acid synthesis. One such drug is acyclovir, I can't pronounce that, which impedes herpes virus replication by inhibiting the viral polymerase that synthesizes viral DNA. So there's antiviral drugs that inhibit the viral polymerase that synthesizes viral DNA. Similarly, azithromycin, AZI, curbs HIV replication by interfering with the synthesis of DNA by reverse transcriptase. So there's also antiviral drugs that interfere with the replication by interfering with the synthesis of DNA by reverse transcriptase in retroviruses. And then it talks about emerging viruses and all the pandemics that have happened that are worse even. It says this virus appeared in HIV, the AIDS virus, is a classic example. This virus appeared in San Francisco in the early 1980s, seemingly out of nowhere, although later studies uncovered a case in the Belgian Congo in 1959. The deadly Ebola virus, recognized initially in 1976, in Central Africa, is one of several emerging viruses that cause hemorrhagic fever, an often fatal syndrome set of symptoms characterized by fever, vomiting, mass bleeding, and circulatory system collapse. A number of other dangerous emerging viruses cause encephalitis, inflammation of the brain. One example is the West Nile virus, which appeared in North America for the first time in 1999 and has spread to all 48 contiguous states in the United States. In April 2009, a general outbreak or epidemic of a flu-like illness appeared in Mexico and the United States. The infectious agent was quickly identified as an influenza virus related to viruses that caused the seasonal flu. This particular virus was named H1N1 for reasons that will be explained shortly. But then it talks about infecting over 600,000 people, killing almost 8,000. Just the influenza virus. And it also talks about the 2009 pandemic, H1N1 influenza A virus. The screening for the flu. 1918 flu pandemic. Many of those infected during the worst flu epidemic in the last 100 years were treated in large makeshift hospitals, such as this one. I can't find it right now, but the Spanish flu. Yeah. The Spanish flu pandemic of 1918 to 1919 killed about 40 million people, including many World War I soldiers. So, why are we making such a big deal about coronavirus that has a 90% survival rate when there was a Spanish flu in 1918 and 1990 that killed 40 million people? And it says RNA viruses tend to have an unusually high rate of mutation because errors in replicating their RNA genomes are not corrected by proofreading. Some mutations change existing viruses into new genetic varieties, strains that can cause disease even in individuals who are immune to the ancestral virus. For instance, seasonal flu epidemics are caused by new strains of influenza virus genetically different enough from earlier strains that people have little immunity to them. So it's saying, like, because RNA has such a fast, high rate of mutations, they can form new varieties of strains that can cause disease even that can cause disease even in individuals who are immune to the ancestral virus. So it doesn't matter if you're immune to it or if you got a vaccine and got immunity. It doesn't matter because it's seasonal and mutations lead to new strains and you can get infected no matter what. And they're trying to completely stomp out the virus, and there's no way to do that. You can't completely eradicate a virus. It'll continue to infect people no matter what we do. Animals that harbor and can transmit a particular virus but are generally unaffected by it are said to act as a natural reservoir for that virus. So there's a replicative cycle of a virus can use an animal as like a middle ground between, they're like a third source of new viral diseases in humans is the spread of existing viruses from other animals. Scientists estimate that about 3/4 of new human diseases originate in this way. Animals that harbor and can transmit a particular virus but are generally unaffected by it are said to act as a natural reservoir for that virus. And it's talking about how the 2009 flu pandemic mentioned earlier was likely passed to humans from pigs. That's why it was called the swine flu. And this is the definition of zoonotic disease. Zoonotic disease is a disease caused from a viral transmission from an animal to a human. And viruses can mutate, so viruses passed from animals to humans can cause new diseases in humans. And it's like the Wuhan laboratory, there was a wet market close by the Wuhan laboratory. The name identifies which forms of two viral surface proteins are present, hemagglutinin H and neuraminidase N. There are 16 different types of hemagglutinin, a protein that helps the flu virus attach to host cells, and 9 types of neuraminidase, an enzyme that helps release new virus particles from infected cells. When an animal like a pig or a bird is infected with more than one strain of flu virus, the different strains can undergo genetic recombination if the RNA molecules making up their genomes mix and match during viral assembly. Pigs are thought to have been the breeding ground for the 2009 flu virus, which contains sequences from bird, pig, and human flu viruses. Coupled with mutation, these reassortments can lead to the emergence of a viral strain that is capable of infecting human cells. Humans who have never been exposed to that particular strain before will lack immunity, and the recombinant virus has the potential to be highly pathogenic.